Hello, in this video we will discuss about the super pathway, methanogenesis, biochemistry. We will learn about the methanogenesis. So what is the methanogenesis? First of all, we will understand about what is this. So basically the methanogenesis occur in water, environment also, and cold sediment and hydrothermal vents as free living cells and as symbiosis with protist and animal that facilitate the methane production and as symbionts with bacteria that promote the anaerobic methane production. It means anaerobic mean without oxygen, not consuming the oxygen and in the presence of carbon dioxide. So remember that things. So in this video we will understand about the human which that contain a microbiome in their gut, in their large intestine, in the cecum. Which that is cecum is the initial part of the containing ascending colon is here near the appendix which that contain bacteria used for the fermentation of the resistant starch. So in this way here is the methanogens. It is the prokaryotes are present a lot of type species. And in this way the anaerobic condition which they require, and in this way the starch as well as other products will convert into the methane gas. So this is the bacteria which that contain DNA as a genetic material, and it is basically prokaryotic cell DNA containing enzyme which support to methane production. So a lot of genes which that produce the different type of enzyme for methanogenesis. So in this case, when we will eat some food containing starch and alcohol drinking, which that go into the stomach and digestion will begin, and the starch will digest in the oral cavity partially and go into the small intestine to the cecum and the ascending colon to the transverse colon to descending colon and sigmoid colon to rectum to anus. So in this way, when going to the large intestine in the cecum, even whole large intestine, the colon, which that contain a microbiota. This microbiome, which that is containing different type of bacteria, used for the symbiosis, helpful for itself as well as helpful for the person because it produces the short chain fatty acid and other products vitamin K, and which that is basically used for our health. So this is the epithelium of the large intestine, the microscopic view. So the undigested resistant starch alcohol will consume by microbiota, and this resistant starch consumed from the microbiome, the microbes which that convert into the methane. So for example, the ethanol or methanol, which that is the alcohol also convert after the drinking alcohol. So the growth of the methanogen will increase due to the ethanol consumption chronically. So this is the acetobacter will produce the acetate, and this acetate will under the condition of the methanogens which that use in the presence of ATP to and acetate kinase which that produce from the gene ACK 2.7.2.1 produce acetyl phosphate. So this is the basically is the yellow star denote molecular marker developed in this study for which sets of PCR primer were designed. So remember that things. And in this way, the presence of methanol, MTAC protein co-methyl transferase enzyme transcribed from the MTAB 2.1.1.90, which that is produce the alpha methyl co 3-methanol specific corinoid protein. So in this way here is you can see this is the CDHC1 for the acetyl phosphate come from the acetate. CDHC1 protein gene will produce the acetyl CoA corinoid protein co-methyl transferase, the co-forming, which that will be carbon monoxide forming will co-forming will produce the 5-methyl tetrahydrosarsenaptaren, while on the other hand the alpha methyl co 3-methanol specific corinoid protein in the presence of mitochondrial protein co-methyl transferase enzyme, which that is used to produce the methyl co. So remember that things. In this way, here is the carbon monoxide also split and in the presence of gene CDHA1, B1 and the carbon monoxide dehydrogenase enzyme to produce the carbon dioxide, and this carbon dioxide will ultimately go under the further process to formyl methanophorane dehydrogenase enzyme in the presence of carbon dioxide and FWDABCDEF, which that will be converted into the formyl methanofurane. So the formyl methanofurane, and in the presence of FTR 2.3 1.101 H4-MPT formyl transferase enzyme used to produce 5-formyl tetrahydromethanopitarine. 5-formyl tetrahydromethanopitarine in the presence of MCH gene and methyl H4 MPT cyclohydrolase enzyme to produce the 5-10 methyl methynyl terahydromethanopterine. This compound will further go into the H2 forming methylene H4-MPT dehydrogenase enzyme in the DMD 1.12.93.2 gene, which that produce the 5-10 methylene tetrahydromethanopterine. This is the compound will generate, and this is the super pathway from the carbon monoxide to produce the methane. Remember that things. So in this way the methyl H4-MPT methyl transferase enzyme to MTRABCDEFGH gene will produce the methyl COM. This methyl COM is basically the methyl COM reductase enzyme, and here is a red star is showing that the MCRABC MRTAB gene for the MCRA gene encoding subunit alpha of methyl coenzyme M reductase 1, and in this way to produce the methyl coenzyme M reductase 1 will produce the basically methane, while on the other hand the methylamine, dimethylamine and trimethylamine will also use in the presence of the gene MTBA will lead to produce the methylated MTMC, MTBC, MTTC and protein basically the coenzyme M reductase 1 will convert into the methyl transferase enzyme to produce the again that things the same methyl COM. And in this way the methyl COM is the metabolic intermediate. In this way the methyl COM here is you can understand the super pathway of methanogenesis, you see numbers for the particular enzymes are shown in parentheses. The red star indicates the MCRA gene, encoding subunit alpha of methyl coenzyme M reductase 1, which is commonly used as a molecular marker for detection of methanogens. The yellow star denotes the molecular marker developed in the study for which sets of PCR primer were designed. So in this study, that is the article, and you can understand the super pathway methanogenesis research paper, which that will indicates by this. The yellow star denote molecular marker developed um, in this study for primer were designed. So for understanding the better the gene transcription and translation. So here you can see this is the red star and yellow star for identification. So remember this a super pathway methanogenesis, the biochemistry we had discussed about. So the same mechanism for the production of methane, but you can understand with the overview. The methane gas will produce in the large intestine to release as a fart during the person, and this is inflammable, and this methane gas is basically hydrocarbon. So thanks for watching. Please make sure to subscribe.